Welcome, welcome in the fourth video lecture of the viral transduction. So in the first video we have discussed about the introduction of viral transduction, why viral transduction is necessary, and in case of which kind of cells we are using viral transduction. And in that video we have also discussed about the advantage and disadvantage of viral transduction, and also we have discussed about a standard protocol for the viral transduction, right? In second video lecture we have discussed about the key features of the viral vectors necessary for viral transduction, right? And in third video lecture we have discussed about the first important viral vector that we are using for viral transduction, which is the adenovirus vector. Okay, so remember, in adenovirus vector we have ended our lecture with a problem that adenovirus cannot express the transgene stably, right? Because the reason was it expresses the transgene in the nucleus episomally, right? That means it is not integrating into the host genome, and that was the problem why adenovirus provides a only transient expression, right? So in this video lecture we are going to discuss about retrovirus based vector. That vectors will solve the problem. Let's see how. So retrovirus is a positive sense single stranded RNA genome containing virus. That means it is a positive strand RNA virus, and it stably integrate their genome into the host cell chromosome. And as it is capable of integrating it with the host chromosome stably, that means it have the ability to stably express, I am referring stably expressed as AC in the host cell, right? So now I want to mention you that retrovirus is a specific type of virus. That means it can infect only specific type of cells. Okay, so if you want to make it broad cell tropism, that means if you want to make it a viral vector that can transfect a wide range of cell, or broadly saying any mammalian cell types, then you have to apply the pseudotyping. So you will apply the pseudotyping with the help of a envelope that will make them the vector that have the ability to transfect any kind of mammalian cell. So here the envelope that we are using is vesicular stomatitis viral glycoprotein. Okay, so that is the reason why this virus can enter virtually any mammalian cell type after that pseudotyping. Okay, so now let's discuss about the disadvantages of retroviral based vector. So the main disadvantage of retroviral based vector is it can only transfect those cells where nuclear membrane is broken down during cell division. Okay, that means we have to rely on only dividing cell to transfect them with the help of our retrovirus. That means retrovirus can only transfect dividing cell. We cannot transfect any kind of non-dividing cell with the help of retrovirus. For example, let's see what I have written here. However, most retrovirus depends on However, most retrovirus depends on the breakdown of the nuclear membrane during cell division, and are thus limited by the requirement of the replicating cells for transduction. Okay, that means only dividing or replicating cells are capable of be transduced with the help of retrovirus. And other disadvantage is it has the power to induce potential insertional mutagenesis. Okay, and we have discussed about what is mutagenesis in our second video. I will put the link on the description box. If you want to know about insertional mutagenesis, then you can consult that video. And the potential for the activation of the latent genes, it can also have the ability to activate latent disease. Latent disease means disease which are hidden, inactive or dormant due to the presence of our immune system. Okay, so and like adenovirus vector, we have seen it can have the ability to carry a gene of 8 kb. Okay, so we have learned that retrovirus is a positive sense RNA virus that have the abilities to stably integrate their genome into the host chromosome. That means it have the ability to stably express the transgene. Right now, the retrovirus we are discussing are specific virus. That means it have the ability to target only specific cells. So if we want to make it broad tropism, so we have to use vesicular stomatitis viral glycoprotein that will envelop them, so that any kind of mammalian cell can virtually be transfected with the help of retrovirus. Okay, and however, most retrovirus depends upon the breakdown of the nuclear membrane during cell division. That means only the replicating cell can be transduced with the help of our retrovirus. Any kind of non-dividing cells are not be transfected with the help of this method. And other important disadvantages are it have the ability to potentially transduce the insertional mutagenesis. Okay, and the potential for the activation of the latent disease. And like other adenovirus, retrovirus have the gene insertion size of 8 kb. Okay, and it has a size of 7 to 11 kb. That means among 7 to 11 kb we are incorporating 8 kb. That means here the dominant part is our transgene. So maximum titer of the viral particle can be 1 into 10 to the power 9, and it is a disadvantage that it can only transfect dividing cell, and expression is stable. That is also an advantage. The problem that was we have faced in case of adenovirus vector, and drawback is the stable potential of the induction of the insertional mutagenesis. Okay, and along with I want to include another point, that is the activation of the latent disease. Okay, so that was the concept of retroviral based vector. So just recapitulate one more time with me. Retrovirus is a positive strength RNA virus that can integrate their genome into host chromosome. That means it have the ability to provide stable expression. Okay, now the retrovirus is have the ability to only transfect specific type of cell type. From that feature, if we want to make them broad specific, what we have to do, we have to pseudotype them with the help of a type of envelope called vesicular stomatitis viral glycoprotein, so that it can transfect any mammalian cell types. Another important disadvantage is it can only have the ability to transfect replicating cells or dividing cells. Okay, and it has the ability to induce a potential insertional mutagenesis. Okay, and it have the ability to activate latent disease. And like other adenovirus based vector, retrovirus have a foreign gene insertion size of 8 kb, and size, that is a genome of 7 to 11 kb, and among them we can include a 8 kb of DNA insert, and maximum titer is 1 into 10 to the power 9. Infection only can be possible for dividing cell, and expression is stable, and drawback is potential for the induction of the insertional mutagenesis, and it can activate any kind of latent disease. Okay, now so that was the video regarding retrovirus. So I hope this video will be helpful to you. Thank you.